Hello, everyone, and welcome to Pocket Size Bio with Dr. Casco. In today's video, we're going to talk about Chapter 4 of Campbell Biology. And Chapter 4 is Carbon and the Molecular Diversity of Life. It is broken down into these three sections. 4.1, Organic Chemistry is the key to the origin of life. 4.2, carbon atoms can form diverse molecules by bonding to four other atoms. 4.3, a few chemical groups are the key to molecular function. As you see in this little photograph here, figure 4.1, the kingling golden snub-nosed monkeys, I hope I'm saying that right, and other living organisms in this mountainous forest in southwest China are made up of chemicals based mostly on the element carbon. Of all chemical elements, carbon is unparalleled in its ability to form molecules that are large, complex, and varied, making possible the diversity of organisms that have evolved on Earth. Okay. So what does carbon have that makes it so special? Some key things we're going to talk about is that carbon can form four bonds. It can bond to other carbons, and it could bond, of course, to other atoms. Commonly, it bonds to hydrogen, oxygen, and nitrogen. Okay. And some of these interactions create chemical groups. Okay. And carbon being able to bond to itself can create carbon skeletons, right? As you see here, these black little balls, of course, represent carbon. And in this case, you have a very special or very specific molecule called dopamine. It is used, it has many functions. As you see here, here you have parenting function and bonding between the mother and the infant, right? It's a signaling molecule. Okay, so let's get to it. Concept 4.1, right? Organic chemistry is the key to the origin of life. Let us go to my notes. Okay, just let me zoom in here. All right, so just some concepts, vocabulary that we're going to get into, and ideas, right? So, definition of organic chemistry. This chapter does deal a lot with organic molecules, and the study of organic chemistry is based on carbon-containing compounds, right? And then there's simple molecules like methane, CH4, to large biomolecules like proteins. Generally, we're going to see this a little bit further down. All these molecules have carbon as a base where it builds upon and creates the overall structure and a function of that particular molecule. All right. So the idea that life arose, right, from having organic compounds from an environment in the early Earth that didn't have any organic compounds was came about from the experiments of Stanley Miller. Okay, in 1953, he investigated the abiotic synthesis of organic compounds. Okay, these are quite complex compounds in order for life to form. So how did these compounds come about? Okay, so this experiment that Stanley Miller did gave the notion that from inorganic compounds, you can create organic molecules, right? Supporting the idea that organic molecules could form spontaneously under Earth's condition. Just to provide a little bit more context, abiotic synthesis and the origin of life. Experiments suggest that organic compounds could have formed near volcanoes, playing a role in the emergence of life. Can't have life without organic compounds first having formed. Okay. And one of the interesting things that astrobiologists will look into if there's carbon-based compounds on other planets, right? Like Mars. NASA's Curiosity rover in 2018 found carbon-based compounds in a crater, raising the possibility that life or ancient life has appeared or been in Mars. Okay. So to have these organic molecules in general, you have these elements, right? Carbon, hydrogen, oxygen, nitrogen, sulfur, and phosphorus, common in many life forms, right? In biology. And that also gives a sense of evolution. We share that similar idea of structure, molecular structure that we are all composite of or composed of. Okay. All with the idea that carbon is so versatile. It's able to form four bonds. So you can have a lot of diversity of organic molecules. Okay. I mean, if you have a lot of diversity in organic molecules, then you ultimately have a lot of variety on life. Okay. And in this inquiry, little panel here, there's a little description about Stanley Miller. It is important to know why this experiment was so fundamental to the understanding of origin of organic molecules, right? So in 1953, you have Stanley Miller um, in a closed system, right, to mimic the conditions thought of at that time in early Earth, right? So you have a flask of water simulating a permeable sea. Okay. So we'll start here. So you have this flask and you have this volume of water, right? H2O. And, water is heated so that it becomes vaporized. And that vaporization of water will simulate an atmosphere. Okay. So that vaporized and water will be vaporized and moved to a second higher flask, this one that you see here, right, containing basically an atmosphere of a mixture of gases. He would provide electrical discharges, right? Sparks would discharge, basically simulating lightning. Okay. So you have number one, water mixture in the sea flask was heated, vapor would rise, go into this atmospheric chamber. It would have a mixture of gases, hydrogen gas, methane, ammonia, water vapor, H2O. And then you would have this form of energy, right? These sparks in this atmosphere simulating lightning. And then whatever condenses back into a liquid form, okay, simulating rain, would come down containing these organic molecules, which would then be deposited into this chamber. A condenser cooled the atmosphere, raining water would come down, get collected, right? In this sea flask here. And that would be the sample of chemical analysis that he would observe. And he found that there was some form of chemical compounds that were organic compounds. Okay, so results. Miller identified a variety of organic molecules that are common in organisms. These included simple compounds such as formaldehyde, hydrogen cyanide, and more complex molecules such as amino acids, and long chains of carbon and hydrogens known as hydrocarbons. So quite incredible and elegant experiment for him to try to collect data, right? Simulating the early Earth and how we can get a rise of organic molecules, because organic molecules is essentially the first step to, the synthesis of life. Okay, may have been synthesized abiotically on early Earth. Although later evidence indicated that the early Earth atmosphere was different from the atmosphere used in Miller in this experiment, recent experiments use a revised list of chemicals also produce organic compounds. Okay, so quite interesting that, in a nutshell, abiotic compounds can, inorganic compounds can produce organic molecules. Okay, 4.2. Let's move to the next section here. Carbon atoms can form diverse molecules by bonding to four other atoms. This section, I primarily talk about just a little bit of characteristics of atoms of carbon. Okay, let me just zoom in a little bit. So the key to carbon chemical characteristics is the electron configuration. Remember, we talked about in chapter 2, the outermost electrons of the electron shell. You have these valence electrons. Carbon has 4 valence electrons in its outermost shell. It can hold a total of 8 electrons, okay, via covalent bonding. right? Covalent bonding, you share electrons. An interesting concept about covalent bonding is that carbon can form single bonds, double bonds, and even triple bonds, where you have that sharing of electrons. Because of this ability that you can create these amount of bonds and these different types of bonds, double and single bonds, as you can see here, you can create different structures, different arrangements based on what you're bonding with. So carbon can produce different arrangements based on single or double bonds and angles. Okay. Of course, based on the space filling model and the VSPR, electron repulsion theory. Okay. Um, so if you see here, right, this is an example of different carbon or organic molecules. Here you have methane, CH4. Okay. You have this central, I'm going to zoom in here a little bit. You have the central carbon, single bonding. Okay. You have single covalent bonds to four hydrogens, and it creates this very nice tetrahedral shape. Okay. Very nice. You have ethane, C2H6. This is a molecule, right? Having a molecule may have more than one tetrahedral group of single bonded atoms. Okay. So you have two carbons in this case. If you notice, still the four bonds that carbon can do are always filled, right? And then you have these two tetrahedrals facing each other, right? Ethane, a different structure, different 3D arrangement there. And then you have ethene or ethylene, right? When two carbon atoms are joined by a double bond, all atoms attached to those carbons are in the same plane and the molecule is flat. So quite interesting, just by creating a double bond between the two carbons, you're creating a different structure, of course, but you're creating a different 3D um, um, structure also. So here you have tetrahedral structures. Here you have a planar structure, right? Flat plane. Okay. And again, that would produce different properties of that molecule. Okay. Let's take a look here at carbon dioxide. This is another example. You have uh, um, a central carbon here, bonded, double bonded to two oxygen molecules. Okay. Carbon dioxide. It is compounds containing carbon are considered organic. There are some exceptions though. Um, Carbon dioxide is an incredibly simple molecule. It lacks hydrogen. Generally, organic molecules would also have hydrogen. Okay. So the idea being still, living organisms thrive on carbon compounds. Urea is an organic compound found in urine. This is an example of organic compound. Okay. And again, as I mentioned before, carbon can bind to many, many other carbons to form chains, right? And you'll have this carbon chain here. They're also known as carbon skeletons. Okay. And again, this is just a review of different relevant atoms that bind to carbon, nitrogen here, oxygen and hydrogen, each with their ability to form several bonds, right? Number of bonds that the elements can form, four, having the highest potential, if I can say, three for nitrogen, two for oxygen, one for hydrogen. Okay, let's move on to the next section here. All right. 4.2.2. Molecular diversity arises from variation of carbon skeletons. Okay, so going a little bit deeper into that 3D structural formula of carbon and the skeletons it can create. So, carbon chains, they form a base of most organic molecules. And this carbon chain, this carbon skeleton, can have different forms. You can have straight, branched, or even ringed structures. Okay. Aside from the shape, there are double bonds that could be present within the structural formula, right? And you can have several of these throughout the skeletal structure. Okay. And again, Depending where you are, you'll have some more complexity in the molecule, different functions, and so forth. Again, as I mentioned before, skeletons can also include other elements, nitrogens, oxygens, hydrogens, of course, phosphorus. To see a visual here, all right, here's a straight um, carbon chain. You have ethane, two carbons, nothing complex. You have propane, three carbons here, right? But you could have a multitude of carbons chained together. Okay. This is a quite interesting one where you have carbon, four carbons in a chain here. All right. You have butane. And that those same, the four having just four carbons, you can have a different structural organization, right? One, two, three, four carbons, but now you have this arrangement here. This is butane, and this is isobutane. Okay, just because of the arrangement of the carbon skeleton. Okay, and then you can also have, let's say you have the straight chain of carbons, and then embedded within this carbon chain, you can have double bonds. You have one butene, you have two butene, depending on where the double bond is found. And again, you can have slightly different properties or slightly or more varied properties. Again, the carbon skeleton can also form rings, very, very common in biology and biological molecules. Okay, where you have the carbon skeleton bonding eventually from the first carbon, the last carbon will bond together. You have cyclohexane in this case, and even within rings, you can have double bonds embedded throughout um, the ring structure. This is a very famous molecule, benzene, where you have double bonds in every other carbon bonding, as you see here. Okay, so variation in structural organization there. All right, getting into a little bit more specific specificity. Okay, you have hydrocarbons. Just let me underline this. Hydrocarbons are basically molecules consisting of carbon and hydrogen, thus the name. It's a major, major component of petroleum and fossil fuels, right? And this is material produced from past organisms that have decomposed, and their carbon content has been processed into these compounds, right? So these hydrocarbons can create a variety of molecules. Okay. Some molecules have regions of carbon and hydrogen like fats, as you see here. This is a micrograph of fat droplets, okay, in fat cells. And within this fat, if you were to zoom in, you would see a fat molecule here. And if you see this carbon chain, okay, whoops, and you have the little hydrogens bonding to the carbons there. And there's three hydrocarbon chains in a fat molecule. We're going to get into um, fats a little bit later. Okay. Now, usually when you have a saturation and when you have carbon and hydrogens in this form, right, they're a little bit difficult to dissolve in water. They're hydrophobic, they're non-polar, okay, and they could release a lot of energy. Right. So if you remember being hydrophobic, being hydrophilic, okay, non-polar, they have no charge, and so forth. Again, we'll come back into this in a little bit more detail. Isomers. Isomers are variations in the structural arrangement of organic molecules. They have the same molecular formula, okay, but they have different structural variants. Okay, so um, they're referred to as isomers. Different structure. If they have a different structure, then most likely they can have a different function. And there's three different types of isomers that we're going to take a look at here. So the first one, structural isomers, they differ in covalent arrangement. You have branch versus linear, as you can see here. I'm going to move my little visual video here. Sorry about that. Okay, so here you have just a straight chain, five carbons, pentane. And here you have one, two, three, four, five carbons, also 2-methyl butane. Okay, so they're structural isomers. All right, they can also differ in the bond location where they're branching out. Okay, you can also have cis-trans isomers. They're referred to as geometric isomers. They differ in the spatial arrangement due to inflexibility of double bonds. Okay. And I'm going to show you the visual there. Cis has an atom on the same side. Trans, they have it on different sides. And this can have a dramatic effect just on the arrangement on function. Okay. Retinol, a, um, a, a, uh, a molecule, right? Trans fats, another molecule we're going to talk about. So what I'm mentioning here, right? You can have basically a functional group here, let's say, where you have this functional group um, on the same side of this molecule, or you can have them in opposite sides of the molecule. All right. So this would be a trans arrangement. Let's say this is an OH and this is an OH. Okay. There'll be an opposite sides. Here you have an OH and then you have an OH here. Right. So this would be cis. Same and different sides. We're going to get back into cis-trans also in a bit. Enantiomers, right? These are mirror images of molecules. They differ in shape due to their asymmetric carbons. It's very nice that they give the analogy of your left and right hand. Enantiomers, due to um, an asymmetry in the carbon, you do have a certain type of orientation, right? And here, because of that um, orientation, you have two different forms, right? You have left, and uh, it's not really left, but it's referred to as an L isomer and a D isomer. Not shown here in the textbook, level rotatory and dextro-rotatory isomers, right? But in essence, they're mirror images to each other. Okay, I'm going to continue with the notes just for you to have here. Enantiomers are left-handed or right-handed versions of the molecule. As I mentioned before, the glove or the hand analogy. Generally, only one version is biologically active, especially in biology, right? Very, very important in terms, especially when you want to focus on pharmacology, right? Drug discovery and things like that. Um, Due to the efficiency of the structure, and you have some um, different effects just on the orientation that it's having. Ibuprofen versus albuterol, methamphetamine versus a nasal congestion medicine. So organisms and your biology, your molecular biology is so specific and so sensitive that even this tiny difference can be detected, right, within living systems. Hey, here's a little panel just to give you an idea pharmacological importance of enantiomers. You have ibuprofen, all right? It reduces inflammation and pain, sorry. In this case, you have the effective enantiomer, and then you have its mirror image, or ibuprofen, that does not do anything at all. Same thing with albuterol. It relaxes bronchial muscles, improving airflow for asthma patients. You have R-albuterol, right? This specific orientation. S-albuterol has no effect. So it is incredibly interesting how such a difference can be all the difference, right, in medicine. All right, let's go to 4.3. So in terms of 4.3, we talk about a few groups are key to molecular function. You will become familiar with these groups as we go along throughout the chapters. Some of them you'll see more often than others. Okay. But in general, right? So I talked about the carbon skeleton, and the carbon skeleton has different atoms that can join to it, but it also can have a chemical groups, right? And chemical groups are small little subgroups of different elements, right? Or atoms, right? That can give the overall molecule a specific property. Okay. So the function and properties of an organic molecule depends on the carbon skeleton and the chemical groups that are embedded in it that are attached to it. Okay. Here you see an example of two different molecules, estradiol, testosterone. Okay. These belong to testosterone belonging to a group of steroids. So you have this ring structure, right? And you have these little groups that are sticking out of this ring structure. And just by having particular groups embedded within this ring structure, you have different properties. Okay. And then here you have CH3 and you have this double bonded oxygen. Here you have an OH group bonded to the structure here. Just by having these chemical groups, you can have different properties. Let me go down, and here's a little table for you to see on the chemical groups. So, impact on molecular function, chemical groups can directly participate in reactions or influence molecular shape. Very, very important. Okay. So, they contribute to, yeah, the molecule's unique characteristics. Okay. And there's key chemical groups that are essential to life and life processes because they modify molecular behavior. Okay. As I mentioned before with estradiol, which is basically estrogen, and testosterone, right? These are both steroids and they have very similar carbon, a carbon skeleton, but they differ in the attached chemical groups. Again, this will lead to different biological functions. So quite important. Okay. In terms of chemical groups and sexual characteristics, just to go a little bit deeper into that, right? So you have these estradiols and testosterone. It would basically give distinct male and female traits in vertebrates, right? And that will alter the overall molecular shape and function. Okay. So these chemical groups, they're often called functional groups, right? Because they can participate in reactions, in chemical reactions. Okay. There's 7 key groups. You should start to familiarize yourself with them. You should memorize them, actually. Um, the most biologically important, you have the hydroxyl groups, the carbonyl groups, the carboxyl groups, amino, sulfhydryl groups, phosphate, and methyl groups that you see here in this table. Okay. You should memorize. You should be able to identify them, right? If you see a visual there. Hydroxyl groups, they basically provide the organic chemical group of alcohols, right? Um, basically an OH group. The carbonyl groups further classified into ketones and aldehydes, right? Aldehydes. You have ketones. Generally, it's a C double bond, a carbon double bonded to an oxygen. Generally, the ketones are when you have this carbonyl group within the carbon skeleton, and the aldehydes you have them when they're at the end of the um, of the carbon skeleton of the molecule that you're talking about. Oops, sorry about that. You have carboxyl groups. They generally act as acids. They donate hydrogen plus ions, right? They covalently bond to oxygen and hydrogen, and that covalent bond is so polar, so it can donate a hydrogen plus ion. Generally, it's in this ionized form, right? This carboxylate, but it's an organic acid. For now, this is good to know. Do memorize, visually the chemical group. OH, a carbon double bonded to oxygen. Here you have the same thing, but you have an OH group, OH bond to this carboxyl group. You have an amino group now. This acts as a base, very, very important for amino acids. Okay. Um, Yes, and they're referred to as amines. This is an example of glycine, an amino acid. You have a sulfyl hydryl group. Usually the compounds that are having um, this SH are thiols. Cysteine is an example. A phosphate group. We're going to take a look at this heavily with DNA and ATP and so forth. But in general, this is a phosphate group, organic phosphate. And then you have your methyl group, right? CH3. And it's basically any methylated compound that contains this would be a methylated compound. Okay. Very important, especially for epigenetics. We're going to take a look at that later on. Okay. So, sorry. Chemical reactivity and solubility. 6 of these groups are generally reactive. All but sulfyl hydro are hydrophilic, enhancing solubility. So they're quite soluble in water. Okay. Methyl is interesting that it's non-reactive, but it serves as a biological marker, especially in DNA. Depending on the pH that you're in, some groups like carboxyl and amino groups, the amino groups can exist in ionized form under cellular conditions, and this has a lot of implications with chemical reactions. Okay. Okay, and the last thing, again, as I mentioned before, is phosphates, phosphate groups, and ATP. We're going to visit this in incredible detail when we talk about cellular metabolism. ATP is referred to as adenosine triphosphate. It consists of adenosine attached to 3 phosphates. As you see here, you have 3 phosphate groups attached to adenosine. And then when ATP reacts with water, 1 phosphate group will hydrolyze and split off, right? As you see here, ATP reacts with water, and you have this inorganic phosphate detached to the molecule, and this will liberate energy. Very, very important for cell respiration and photosynthesis. Okay, so the removal of phosphate forms from ATP releases a lot of energy, right? Which can be used by cell and by cellular processes. So ATP stores potential energy. Sorry about that. Okay. Not energy itself, but potential energy. Okay. So just a little bit of review here. Review of the chemical elements of life. Living organisms are primarily made up of carbon, oxygen, hydrogen, nitrogen, and smaller amounts of sulfur and phosphorus. Okay. Covalent bonds is a strong chemical bond. It is very crucial to be the basis of all these molecular compounds because carbon can create 4 bonds, right? And can bond to itself. There's a lot of diversity that can occur, okay? In terms of biological structures. All right. So that's it for this chapter, guys. Thank you very much for listening, and I'll be back with another one. Take care.